Well, you see that the presentations we've had today have covered a whole gamut of, let's do it, Scott, of possible interventions in Parkinson's disease. And so we moved from conventional medicine into lifestyle interventions that Dr. Tudor-Locke has demonstrated. And I'd like to move into another area, which is nutritional interventions for Parkinson's disease. Now, I don't want to pose myself as an expert in this area. This is an area that's new to me. I am an expert in nutrition and brain aging, but just not specifically in Parkinson's disease. And I have been involved in pharmacologic development too, specifically for Alzheimer's disease. But coming to Pennington, I've just gotten very, very interested as an alternative approach to medicine, that, you know, there are things that we can consume and things we can consume more of that may affect the risk of the disease as well as the course of the disease. So we're trying to determine whether there's nutrition that will keep you balanced and moving forward. So you are what you eat, no question about it, and that includes your brain. And we know that some diets can have very detrimental effects on the brain, and we know that some diets can have beneficial effects. But just in the short time allotted to me, I'm just going to ask the question, are there foods and beverages or food derivatives or nutritional supplements that can reduce the risk of Parkinson's disease and or improve function of those who already have the disease? Now, scientists go about answering this question in three very distinctive ways. Each has its strength and all have weaknesses. One is the population studies. I'm going to give you some examples of that, where you just look at large populations of people with the disease and without the disease and determine what about their environment or their genes possibly or relate to the risk of this disease. And then there are preclinical models. This has generally been my area of research. We work in vivo or animal models, or in vitro and cell models. And these you've already seen in Dr. Stannard's presentation today, that you know, cell culture can have incredible effects on screening for drugs. So these are very, very powerful preclinical models. And then finally, the gold standard is going to be a clinical study. You're going to have to take the stuff that you've shown works in animals, that there's a relationship in populations, and now you're going to have to give it to real humans with real diseases and determine through specific outcomes whether it in fact has a benefit. All right, I'm going to skip right through that one just for the sake of time. But anyway, this is a very good review I found that has examined what are called Nutraceuticals, and that means a nutritional product that can have effects like a drug. And this has been a pretty good review. You see the things covered here. I'm going to cover those in addition to two that weren't covered. And this is sort of a little rating system where at least these investigators have looked at these various nutrients, supplements, foods, and food supplements, and determine whether it looks like there's a possible benefit. All right, taking one that I honestly had never heard of before a couple of weeks ago when I started putting this talk together. I knew all the other ones, but this is one that's a very interesting plant. It's called a velvet bean, found in Africa and India. Contains in itself the high concentration of L-DOPA and CoQ10. Anyone ever heard of this? So I want to ask you this a lot, raising your hand. Okay, so I'm just curious. Okay. Yeah, now in terms of animal studies, again, these preclinical studies, there have been a couple of papers done. This is usually animal studies you take and you put a toxin into the brain that destroys those neurons that produce dopamine. So everybody's familiar with those kinds of models. So this was a stepping test. They looked at quantitated the amount of steps that the animal can take. This is sort of quality of stepping. So this animal is the higher the score, the worse the score. But these two concentrations of this water extract of the where did I go here? Yeah, of this interesting plant produced a better performance. And this is a dyskinesia score, and this is a drug is given to produce. It's down here. It's this drug is down here, and it's a dopa decarboxylase inhibitor. So it makes them worse in having dyskinesia. But again, look, it almost disappears with the treatment from this water extract of this plant. And there has been some time ago now a double-blind study. It wasn't a very large study that did have reports of the benefits, if you can read right there, particularly reducing the dyskinesia. So the animal work did translate to the human element. So this is very interesting that there is a natural product that does have L-DOPA and CoQ10. Now, what about vitamin D? I mean, this is really in the newspaper these days. A lot of medical journals, and sort of a rediscovery of this essential vitamin, and probably related to our lack of being out in the sunshine. We just don't. Our bodies are capable of making vitamin D, but we need sunshine to do that. So here you see that the conclusions down here are that individuals with high serum vitamin D concentration showed a reduced risk of Parkinson's disease. And the relative risk between the highest and lowest was about, an order of about 33% difference in risk rate. Now the results are consistent with the suggestion that high vitamin D provides protection against Parkinson's disease. But importantly, this is this is a population study. This is just showing you a relationship. It doesn't mean that if you take more vitamin D, you're going to have a less risk of Alzheimer's disease. So it cannot, however, be excluded that finding is due to residual confounding or other and further studies are needed. That's always how epidemiological studies end up in their conclusions. All right, you're going to see a lot of this. I'm going to throw bottles up there. You know that you can go out to stores and buy this, and a lot of people do. I mean, but that doesn't mean I'm recommending it. I'll reinforce that point throughout. But here's an important element of that bone and mineral metabolism. We've heard of vitamin D3 in relationship to a low bone mineral density. So here's a study. This is a fairly recent study in 2009, and here's the Parkinson's patient, a pretty large sample, 82, and controls. And these are measures on DEXA. A lot of in the audience have had DEXA. This is a bone scan. And so these are measurements of the strength of bone in the femur and in the lumbar. And this is what scientists look for, these p-values. So every one of them were significant, indicating that the bone mineral density was less in the Parkinson's patients. And then when you looked at their vitamin D3 consumption, this is the biochemical measure of it over here, that you can see that the controls, 21 versus 12, were twice as high. So there does seem to be this relationship. Now, Dr. Callaghan's already mentioned CoQ10. This is that trial that was done in 2004 that looked fairly promising. CoQ10 is thought to be sort of a vitamin, but it's more than that. It's sort of a metabolic cofactor, very critical metabolic cofactor, very important in regulating metabolism. But you can see that these are 3 different doses over quite a long period of time, 16 months. And this is a standard endpoint, the Universal Parkinson's Disease Rating Scale. And you see that, like a lot of things, there's a dose-related dependence here. So the symptoms get worse in the placebo-treated group. This is a double-blind study, and the 3 doses, particularly the higher dose, shows improvement in that score. But that's 2004, and he mentioned there's been more recent studies that just haven't supported that. And that's the problem going forward. All right, creatinine. Who's heard of creatine? Creatine. If you have any bodybuilders or gym rats in your family, then they really believe in this. It's for muscle building, but also has effects on brain. There's been some very interesting animal models. But this was a study that was done about 6 years ago, 2006. Again, using that same, this is a change in that score. So it's like a total change in the score. So you see that this is minocycline, which is an interesting antibiotic, but it didn't seem to have any effects compared to placebo here in the dotted line. But there is a slight and significant effects of creatine. All right, how many people take this medication? This supplement, Omega3 Fish Oil. I do for my own metabolic syndrome, a prescribed form of it. And we've learned a lot about, used to be, we went through this phase that all fats in your diet are bad. And now we know that there are good fats and bad fats in our diet. And so this Omega3 is one that does seem to have protective effects in a wide number of diseases. So here we have another animal model, and we can give a toxin. These are the nice dopamine producing neurons in the brain stem of in the midbrain of the animal model, and we give them the toxin. It destroys those neurons, or most of them, about a third of them. So we count those neurons that are destroyed, and we see that we wipe them out, but down to a level of about 2/3 of the level of controls. But if the mice had been fed on this diet enriched in omega-3 fatty acids, that they were completely protected. That's very promising, but again, it's an animal model. All right, ginseng, I'm sure everyone in the audience have heard of that. It's had all kinds of health claims, and a lot of people take it as a supplement. And here we see another animal model. It's the same one where we give that toxin that wipes out those nice dark cells there. That's what are called the substantia nigra. And then we look down here at the cell destruction, which in this particular case was much more complete. They just gave a higher dose, destroyed many more of the cells. But again, in the mice that were fed the ginseng, we see there was incredible protection in this particular study. All right, curcumin. Now, you've heard of ginseng. How many people in the audience have heard of this? This is so-called curry spice. I'm just showing you one example of an in vitro study. So this is just taking the mitochondria. These are the parts of the cell that produce the energy in the cell. And you can isolate those and you can throw a lot of toxins on them. This happens to be a toxin that would kill them, but they're more protected in that in vitro environment if they were given curcumin. Now, you got to go a long way from cell culture studies to real life applications, but I want to tell you that curcumin, I've seen clinical results now in Alzheimer's disease look very promising. It's been used also in cancer. This is a very interesting nutraceutical product. Okay, something we're very familiar with here in this area of teas. I'm just showing you one study in Japanese and Chinese teas. So how many in our audience knew about this relationship between tea consumption and Parkinson's disease? All right, so just going to highlight over here that, so this is showing coffee, and I'm going to say a little bit about that later, black tea, Japanese tea, and these are what are called odds ratios. So over here in the quartile is drinking the highest consumption of these beverages, coffee, Japanese tea. We see the probability is over here, and that means it's significant. So the more tea, the more coffee are being consumed, the less risk of Parkinson's disease. Again, that's a relationship. It does not translate into meaning if you go out and drink a lot of coffee, you're not going to get Parkinson's disease. Okay, here's one we should also be happy about. This is wine drinking. This is not risk of Parkinson's disease, but it's tremor. And this was a fairly large study. It had 83 patients in it. And again, you're just asking a question. How many glasses of wine do you consume? So you have none, you have 1 to 2 a day, and you have 3 glasses of wine a day. Now, I'm not going to ask you to poll the audience, how many people drink 3+ glasses of wine a day. But you can see, not unusual from the epidemiology of wine for other diseases, excuse me, that the risk factor is down to that. That means almost over 80% reduction in risk rate. Again, this is one sample. It's 84 patients, but it's interesting. Now, how many people in the audience have heard of this interesting compound, resveratrol? No, it's associated with wines and with grapes. This is a very interesting, what's called a polyphenol that is found in grapes that helps protect grapes against fungus. All right? But this interest in this compound has exploded. I've been involved in, again, preclinical work in this. It has some very interesting effects. But here we should see another model. This, rather than a toxin, this is a compound that induces inflammation in the brain, and then affects the uptake of dopamine uptake. And you can see that that when you give it, it really knocks the heck out of uptake. And then these are the concentrations of resveratrol that the mice were provided prior to that. I'm sorry, this is an in vitro study. So this is cells. So you give them, treat them with resveratrol, and you get protection against that loss of dopamine. And these are killing those neurons that are making the dopamine. This is without treatment. And here are the 3 doses of resveratrol that shows protection in this in vitro study. Now here's an in vivo study that's very relevant to Dr. Callaghan's presentation, where he showed you that he has to put a probe down inside the brain, and he mentioned there could be some bleeding, there could be some infection. So this is an animal model in which there's a cannula. That's just basically a steel tube that's injected into the brain and creates some damage. And these are the treatments with resveratrol that's looking at the size of the lesion that's made. You want to try to reduce that as much as possible. These dark cells here are inflammatory cells that are called neuroglia and microglia. And you can see that the mice that were treated with resveratrol prior to the surgery have less signs of this inflammation. So this compound is in clinical trials for a number of applications, including Dr. Ravison at our institute just put in an application to do a clinical trial in obese diabetic patients. I'm also at the LSU Med School. We're considering a trial in AIDS patients. This is a very interesting compound, I would say, at the head of most of these applications I've presented to you so far. Okay, coffee. I mean, the good thing about Parkinson's disease, you know, smoking reduces the risk. Again, it's an association. And coffee reduces the risk. Again, it's association. But coffee now has come from being one of those things that are not so good for you to studies, epidemiology, and also real clinical trials has shown it can have these beneficial effects outlined over here across a wide range of function. This now is, you don't have to read the whole thing, but is a very recent meta-analysis that has looked at all the studies that have been done. And down here, just to point this conclusion, this study confirms an inverse association between caffeine intake by way of coffee and the risk of Parkinson's disease, which hardly can be explained by bias or uncontrolled confounding. So they're saying we've looked at a number of studies and this relationship seems to be real. That's led to an interesting connection between coffee and caffeine and its relationship to this set of receptors in the brain that are called adenosine A2A receptors and are actively being investigated in terms of pharmacologic intervention for the disease. Scott, did it freeze up on me again? Yeah, it did. Boy. Whoa. That didn't have any animation in it. Where were we at? Keep going. Scroll down here. Let me see a coffee. Right there. Right here, right here. All right. I'm going to wrap up with one very interesting berry, and actually several berries that I've had some experience in preclinical assessments and also in clinical assessments. And these have been investigated starting back in 1999 in preclinical models, in animal models by my two colleagues, Jim Joseph and Paula Bickford. And a lot of the things I've told you about have to do with these foods and these supplements serving as antioxidants, reducing something that is called oxidative stress. Our bodies produce oxygen radicals that can hurt and harm our own cells. Now we can look at motor performance in animal models in this sort of little rat Olympics you see outlined here. But I want to tell you about this particular task. This is called a rotorod. So the rat is on this cylinder, and this cylinder is rotating, and it gets faster and faster, and they try to stay on, they fall off. We count the amount of time they can spend on that rotating cylinder. It's sort of like being a lumberjack in the middle of a river back in 100 years ago on a log. All right, so you can see that with aging. So this would be a college student, and these are over here retirement age rats, and you can see that the amount of time that they spend on that rod decreases quite dramatically in middle age and is less so at the age I stand in front of you today. All right, but look at this remarkable finding here. This is blueberries and strawberries. This is supplemented to the level about 2 cups a day of these different berries. This is blackberries, and the improvement in these animals is remarkable. They didn't gain any weight or lose any weight. They ate the same amount of food. But there's a lot of interesting changes that are made in their brain from this about 4 to 6 weeks of feeding of this berry-enriched diet. All right, so can foods affect Parkinson's disease? Yeah, I'm serious that they possibly can, but I've only given you a tasting menu. I mean, the menu could be much broader than that. But it is just a menu, and you might come away feeling a little bit not satisfied. Because I want to emphasize, I've given you information, but it is not a prescription. Don't go home and say, I need to go do this, okay? So I would summarize in this fashion. I mean, there are many promising candidate nutritional treatments to consider. I've given you that tasting that those possibilities exist. However, all of these will require additional rigorous clinical testing that will be sufficiently powered, double-blind, placebo-controlled. That's the only way we can know for sure if something works. Now, these are the types of studies that we'd like to generate in the future here at Pennington. So that was the reason for our conference today, and I just wanted to give you a flavor for that. So I want to reemphasize, do not go to your local health food or drugstore and buy any of these products that I've mentioned to you today unless you consult with your physician regarding such decisions. We found like proteins can interfere with the medication that you're taking already. So that's how I would conclude today. I really, again, thank you for your attention, and we'll go on with our next speaker.